Hello. Today, we will be discussing the determination of gender. Many people believe that this is solely a genetic issue, but we will quickly learn that there is a large amount of hormonal play that goes into the development of sexual determination and sexual characteristics. First, we will start off with the basics. Male and female sex organs consist of three sets of structures: the gonads, internal genitalia, and external genitalia. The gonads produce gametes. The male gonads are testes, which produce sperm, and the female gonads are ovaries, which produce the ova. The combination of sperm and ova are what give rise to future progeny. However, there is a time when these cells cannot yet be differentiated as male or female gonads and are simply referred to as germ cells. Internal genitalia includes the accessory glands and ducts that connect the gonads with the outside environment, and external genitalia include all the external reproductive structures. Now, the main factor for determining sexual development in an individual is the human genome. The human genome is the complete set of nucleic acid sequences for humans. In simple terms, an individual's DNA. DNA is easily broken down into chromosomes, which are separated into two categories: autosomes and sex chromosomes. Each nucleated cell body has 46 chromosomes, called the diploid number. Each chromosome is a constituent of a pair, or 22 matched homologous pairs. These diploid cells are referred to as 2N. They are responsible for the development of the human body and its phenotypic expressions. The sex chromosomes are different from autosomes, as they are haploid cells that determine solely the sex of an individual. Being haploid means that instead of containing 46 chromosomes, they only contain 23, and are referred to as 1N or solely just N. When these chromosomes are combined, they create a diploid zygote with 46 chromosomes. These sex chromosomes are separated as being either X or Y. The X chromosome is much larger than the Y chromosome, as it contains a larger amount of coding information. These sex chromosomes are directly responsible for the development of internal and external sex organs. Sex chromosomes are normally distributed, with one coming from the maternal side and one from the paternal side. Genetic females are XX and receive one X gene from each parent. Genetic males are XY and receive one X gene from the maternal side and a Y gene from the paternal side. The Y chromosome is required in order for production of male reproductive organs. However, an abnormal distribution of these genes can take place. This is simply when there is an absence or extra addition of chromosomes, resulting in too few or too many for normal distribution. Examples of this could be XXY, YO, XO, etc. These occurrences result in genetic disorders and/or diseases. However, it needs to be noted that regardless of abnormal distribution, the appearance of a Y chromosome will always create male reproductive organs. So, regardless of how many X chromosomes there are, if a Y is present, the individual will always be male. However, if a zygote only receives a sole Y chromosome, it will die. X chromosomes are needed. Remember, the X chromosome is much larger, and that is because it contains genetic information that is vital for proper development. Because of this, a YO individual will not survive, but an XO individual can. In the beginning stages of development, the sex of an embryo is unable to be determined. It isn't until around the 7th week that reproductive structures begin to differentiate. Up until this point, the embryonic tissues are considered bipotential, as they cannot be sexually identified. A bipotential gonad has an outer cortex and an inner medulla. It is signaled to become a male. The medulla will develop into the testes. However, if signaled to become a female, the cortex will develop into ovarian tissue. An easy way to remember this is for M for male and M for medulla. Those two M's will pair up. However, if there is not male, not medulla, the cortex and female pair up. Bipotential internal genitalia contains two pairs of accessory ducts: Wolffian ducts from the embryonic kidney and Mullerian ducts. One pair will degenerate depending on sexual development of male-female. Bipotential external genitalia consist of a genitalia tubercle, urethral folds, urethral groove, and labioscrotal swellings. These will change as sexual development occurs into either a male or female. The signal to become a male or female is due to the presence or absence of the sex-determining region of the Y chromosome, or SRY gene. The male embryonic development can simply be broken down into these 5 steps. First, the SRY gene produces testis-determining factor protein, commonly referred to as TDF. So remember, once again, without the Y chromosome, that SRY gene will not be there. So the male has to have the Y chromosome. Once the SRY gene produces TDF, the TDF will bind to DNA and activate additional genes that cause gonad medulla to form the testes. Once the testes have formed, they have interstitial cells and Sertoli cells that have formed. The interstitial cells secrete testosterone, which controls development of Wolffian duct into accessory structures and development of external genitalia with the aid of dihydrotestosterone, often referred to as DHT. The Sertoli cells secrete an anti-Mullerian hormone, which causes regression of the Mullerian duct. So, from what we can see here, the Y gene started the process, but the TDF binding to the DNA started the forming of the testes, which started the cells in the Sertoli cells. The interstitial cells and Sertoli cells began releasing hormones. These hormones became incredibly important in the development and regression of the different ducts needed. The same breakdown can be applied for the female embryonic development. First, the bipotential gonad develops into ovarian tissue due to the absence of the SRY gene. Remember, in females there is no Y gene, only X genes are present. The Mullerian ducts develop into the upper portion of the vagina, uterus, and fallopian tubes, and the Wolffian duct degenerates due to the lack of testosterone. Without the DHT hormone, the external genitalia take on female characteristics. Therefore, it can easily be understood that although there are obvious genetic factors that initiate sexual determination, hormones play a very large role. The presence of hormones such as testosterone and the anti-Mullerian hormone initiate male reproductive development and characteristics, while the lack of these hormones leads to female development. I really hope that this breakdown made the determination of gender much easier to understand for you, and gave you a crucial insight on how hormones truly do have such a large factor.